All right, so for our final keynote of Maine 2024, before we get to prizes and goodbyes and parties later, I'm very pleased to introduce Eva Dyer, who's joining us from Georgia. So she's a professor at Georgia Tech, and before that did her PhD at Rice University, and then a postdoc in Chicago at the Rehabilitation Institute of Chicago with Conrad Cording, which is actually where I met Eva. And so I've known her for quite a while. And yeah, as you'll see, I think for those that weren't here this morning, there was a Keynote switcheroo, and the foundation model Keynote is now being done now, and I think you'll now see much of the connections with some of the things that were discussed in that initial panel. So yeah, I'm excited to hear your talk on this large-scale pre-training for neural data.
Hello? Okay, how's that? I feel like I'm in, I don't know, the Matrix or something here. Yeah, thank you so much for the invitation. I'm really excited to be here to tell you about our work in building foundation models for neuroscience. It's also really great to be here in Montreal to talk about this, because it was about 2 years ago that Blake Richards and Joel Zilberberg actually organized a workshop for us to all come together to start to talk about these topics and thinking about, what do we need in neuroscience? How do we build it? What are the data sets? And so really I think of this also as the birthplace of thinking about foundation models for neuroscience. And I'm really excited to present what we've been up to since then. So the motivation of this work is really coming from a lot of the recent advances that we've seen in LLMs and other generative AI technologies that have really kind of opened up a whole range of possibilities that hadn't been possible before. And I think one of the key lessons that we've been able to take away from this is really just the power of scale, right? And what I'm showing here is just some examples from LLM pre-training, where after you get to a certain size of model or you get to a certain amount of data, you start to see these emergent capabilities start coming through in the model training. And really this is because of the fact that when we have enough data and we have a complex enough network, we can start to learn these really complex underlying tasks. And similarly in neuroscience, we've also been amidst this revolution in terms of the amount of data and the types of data sets and the diversity and richness of what we can measure from the brain. And we've seen this really kind of exponential growth in both the sizes of data sets, so how many neurons we can record from, as well as all of the different pieces of brain and where we can go within these neural systems. So this is kind of exciting because it shows us that maybe we are at a time in the development of neural methods and neuroscience where maybe we can also benefit from this scale, right, that we're starting to see. And, this is, I think, a really exciting opportunity, but there's, of course, a number of challenges that make neuroscience data really different from working with large corpuses of text. And in particular, most of when we're building models of neural activity, because each time we see the brain, we see different neurons or different brain regions or all these other diverse aspects, most models or deep learning models that are fit to neural populations and neural activity are typically done one model at a time, right? And so this idea of really being able to benefit from this power of scale will necessarily require us to put all of this data potentially together in a large model. So breaking this kind of one data set to one model perspective. And so really our hypothesis going into this is that neuroscience and neural data analysis could likely benefit from this kind of large-scale pre-training through this sort of unification where instead of having one model for one data set, we have maybe one model that can actually digest and aggregate very large amounts of neuroscience data. So I kind of already alluded to some of the challenges, but yeah, what makes this really hard and a unique challenge is the fact that neuroscience is very heterogeneous, right? So if we're studying the motor cortex or visual cortex, where we're recording in the brain could be different. The way that we stimulate the brain can be very different through different tasks. And then even just the way that we're processing this data can take very different, or produce very different outputs, even if you were studying maybe the same brain region. So this amount of heterogeneity is, pretty unique, and it's something that we have to overcome. And even in the case where we have a very simple setting, where we have like a chronic recording in one part of the brain from one individual over many, sessions or recording over time, even then we're faced with the challenge that the neurons that we get or record from can be changing across days. And we have no correspondence between units that we see across different sessions. And this has caused, and we've heard a number of talks about BCI, BMI applications. This lack of stability makes it quite hard to be able to build like a robust link between the brain and behavior. And so all of these challenges kind of combined, were something that we wanted to try to tackle. And so to do that, my grad student and our collaborators here at Mila, we all set out to build a architecture, something that would allow us to scale and to be able to train on large amounts of neural data. And so I'll go through the sort of architecture and the idea, and then tell you about some of the different ways that we've been exploring using this. Please stop me and ask any questions if you have any at this point as I start to go through the model. Okay, so we've already heard a bit about transformers, and I think this audience is probably pretty familiar with ideas more than others. But when we're talking about transformers or these sequence models applied to text, thinking about how you take a sentence and break it down into words or even chunks of words is somewhat, straightforward, or it's something where we have a pretty good way of, building representations of words and stringing them together into a sequence. And so now the question is, how do we go about taking neural population activity and turning it also into a sequence that we can start to learn from and scale, right? We said we want to do this across many brains that would have different neurons or different underlying functions of those neurons. So the first thing that we set out to do was to build a new tokenization strategy, which would take neural population activity and put it into this kind of sequence level representation. And we do that by identifying that what is the language of the brain, if not but spikes? And so here what we're going to do is we're going to take all of the spikes and all of the activity within a population, and we're going to turn that into a sequence where every token that goes into the transformer is a different spike. This is sort of demonstrated, This is demonstrated in this picture here, where I have 3 different neurons that I'm recording from over time. We can see all the different action potentials or spike times from these 3 neurons. And what we'll do is we're going to take each spike, turn it into a token, and that token is going to have 2 pieces of information. One is some information about the neuron that it came from. This is going to be what we call a unit embedding that's learned over time. So the model can see all the neurons that it's ever seen and try to figure out some way of kind of semantically indexing all of these unique neurons. And that's sort of shown schematically here, where if we were to think about like different words within our sequence as being kind of these embeddings in some high-dimensional space, where maybe words like dog and cat are mapped to similar parts of this latent space or these word embeddings. And similarly, we think that through this form of like implicit alignment, it could be that neurons that have similar function or might have similar properties or ways of interacting with other neurons should also be mapped to similar parts of this unit level space. So that's one core concept, is just this idea that all neurons are given this vectorized representation that's learned, and now the model can find ways of putting together neurons from different sessions, from different animals, or from different brains in a way that's kind of consistent and makes sense, right? And then in addition to having the unit or the information about what neuron the spike came from, we also encode the absolute time of the spike rather than binning the data, which is a more standard approach where you would take a chunk of time, count the number of spikes, and then represent that as like, you know, a binary pattern or integer sort of values. So instead of doing that, we're actually going to take the time at which the spike was emitted, and now through this sort of attention mechanism, we can modulate attention between any two spikes based upon the distance that they are in the temporal space. So for two spikes that happen, you know, back to back, the attention can be modulated by that relative timing differential. So we use a method called rope, which gives us an efficient way of actually implementing this relative position embedding. And in the end, right, we take this potentially thousands of neurons or, you know, populations of neurons, and we've turned them now into this sequence, right, that represents all of the neural activity. Okay, so once we've tokenized our neural data, the next step is to, you know, pass this into a transformer in this case, because we want to be able to leverage, you know, some of the scaling properties that we've seen from these methods. And so, you know, one could imagine, now I have this really long sequence, and I could use self-attention or just a standard transformer to now ask the model to look across all of these different spikes in the population towards solving some downstream task. What I'll talk about initially will be mainly kind of supervised training paradigms where I have neural activity and I want to predict some behavior, right? And so, yeah, so one way we could do is we could pass in this sequence of spikes, put it through the transformer, ask it to predict the behavior at the output. But one of the other key innovations of this method is to basically say, instead of using, you know, full self-attention across this long sequence, instead we're going to use something called cross-attention, or this perceiver encoder, which allows us to take long sequences and basically compress them into a set of latent tokens. The reason why this is really important for neural data is because if I give you a data set with 1,000 neurons or 10 neurons, I'm going to create very different sequence lengths, right? And because we're constantly dealing with highly variable sequence lengths, this gives us a way to kind of compress all of these sequences into a common and fixed set of dimensions. And so this cross-attention mechanism basically works by, so normally in dot product attention, we would compute the queries and the keys over all pairs of tokens. And in this case, we're going to do something different, which is we're going to have a set of latent tokens, right, this fixed bottleneck that I've talked about, and those are going to serve up the queries. And then the input tokens, so the spike tokens, are going to provide the keys and the values. So it's sort of like you could imagine this latent token that might be looking for a motif of activity or certain, you know, patterns of firing, or maybe it's just looking at one neuron that it cares about. It can basically query to all of the input spikes and find the pieces of information that it cares about. And so through this cross-attention operation, right, we're now going to take our sequence, we're going to embed it into this latent space, and then we can apply multiple rounds of self-attention to build like richer features and go further. But now it's operating in this latent space rather than in the original input dimension. And this is also really important for computational complexity, because now instead of working with like n squared, where n is the number of spike tokens, we're working in this L squared space, because where L is, say, the number of latent tokens. Okay, yeah, so these are the other rounds of self-attention. And then finally, at the output, we're gonna do something kind of similar to what we had at the input, in that we have absolute time over which we're going to query from the latent space to generate behavior. So in the same way that we could have irregularly spaced events within our input, we can also query at any arbitrary point in time in order to produce some sort of output behavior of interest. And that's sort of highlighted in this picture here, where we're just arbitrarily querying at these kind of non-regularly spaced intervals. And so, so just like we had the latents providing the queries to the input, now we're going to have the output query to the latent space of the learned representations. And so now those queries will basically be combined also with something called a session embedding, which can kind of capture some unique aspects to different data sets. And then at the output, we can produce our rich behavioral outputs that we were interested in predicting. Here, this is, and what I'll talk about next is an example where maybe you want to predict an arm movement from brain activity. So here we're looking at kind of the kinematics of some reaching movement that we're trying to decode here. Okay, and this is really cool and has some nice properties for just behavioral decoding in neural data in general, because we don't always record behaviors at the same frame rate. Maybe there are events that happen that are irregularly spaced, right? Or maybe you have dense mixed with discrete behaviors. So this kind of flexibility can be really powerful, and it can really cut down a lot of the extra processing as well that often comes with having to temporally align all of your output variables. Any questions? Okay, you got it. Cool. All right. So with that, I can kind of start to now jump into some of the results, experiments, ways that we've been testing this kind of framework for training. And so in a first attempt, we were interested in doing, as I already kind of described, these like motor kinematic decoding tasks that are really frequently occurring in BCI applications. And here we combined a little over 160 recordings or sessions worth of training data from 4 laboratories spanning about 9 non-human primates. And these recordings were coming both from primary motor cortex as well as premotor cortex. Also, the data sets varied in terms of whether or not they were spike sorted or they were coming from threshold crossings. And importantly, all of these animals were actually carrying out really diverse, different reaching tasks and different underlying goals. So some more simplified cases like the center out reaching paradigm, where an animal has to reach towards one of 8 different targets displayed on a screen, has a relatively stereotyped and kind of simplified distribution of movement. But then we also have some of these more rich tasks where you could have self-paced reaches or random targets, where really there's a lot more variability in the distribution of the outputs. And I will say that before we did this, these different tasks wouldn't have been combined into one model or one training data set. So even though this is all motor cortex and reaching, this is actually quite diverse and heterogeneous in that sense. And so, first we kind of we trained on a smaller subset of this data, 100 sessions from a single lab, and across the board we've kind of seen that this notion of scale and pre-training on more and more data seems to consistently improve our ability to decode from held out sessions or from held out trials. And what I'm showing here on the left is results from the center out kind of simpler task, and then on the right is this more complex random target task. On the x-axis is just, so each dot is a session, right, or the R-squared decoding performance from a given session. And we're just plotting here the difference between, what would you have gotten if you just did single session training versus this multi-session training approach. And what we find is, for cases where you either have a limited amount of information, like it's hard to decode from that data set, you get kind of a low R-squared. This multi-session approach benefits you even more, right? Because there was something in the data, but it was hard to learn it from, just that data set alone. And then just kind of summarizing some of these results of scaling, we can just see that, if we have single session models and we take the average over 20 of them, and then what can we do when we now train on all 20 sessions, and now what happens when we add in data from a different task, right? This is the RT task, this more complex one. What if we add in data from a simpler task, from CO, or from this center out? And indeed, we're finding, like, even on a different task, training on, these additional sources of data can be really beneficial in terms of brain decoding. In addition, we have some ability to kind of understand or interpret the relationship or kind of compare different sessions in different neural activity data sets. And so in this case, I'm just showing a PCA embedding of this session embedding that we learn at the output of the decoder that I talked about. And here each point is like a different session, and they're color-coded by the animals. And we are able to now see how many animals are kind of all organized within this same common and unified space. And we do find that animals from the same lab and kind of same recording conditions are indeed clustered. Okay, so what I've talked about so far are cases where we have trained on neurons, right? Like we've trained on all these neurons from all these sessions, and now we have this pre-trained model. And so far I was just testing it on held out trials from neurons that we had seen, right? And the important part about this is, like, with this model, because we're learning these unit level embeddings, we do have to know something about how we're going to put new data from a new brain into this same space. And we have to do that by learning new unit embeddings for the new neurons that we add in. And so we can do this in two ways. One is on a new session, maybe from a new brain that we didn't see during training, we can fine-tune the model to try to either just initialize or update those unit embeddings for the new neurons, holding all the rest of the weights of the model fixed, or we can do a full fine-tuning and also update those model weights as well. And then in both of those cases, we can now test our pre-trained model on an held out animal that that hasn't been seen during training. And this is some of the results of that applied to the neural latence benchmark, which is a benchmark for both unsupervised as well as behavioral decoding from neural activity. And what we found was that, not only does POYO on its own already improve over a lot of the existing baselines for this task, but we actually see that this pre-training and then fine-tuning helps us to go even further, and we're seeing even further benefits. And on the right, we're just looking here at like compute and sample efficiency, so just also showing that we can do this fine-tuning or reinitializing of the units with just a few shots of labeled data. Okay, and just to see how far we could go and how crazy we could be when testing this idea. This is joint work with Patrick Minot and a lot of the folks here at Mila. So in this case, we were interested in, what if we want to test this pre-trained model on a completely different task, right? And maybe even a different species. So here, what we're looking at is the case where, so this is data from a benchmark from Frank Willett, where basically the goal, so a human who's actually paralyzed and locked in, their recordings from their premotor cortex using Utah arrays. And here they're basically imagining writing different letters, and then the goal is to decode that sequence of letters so that you could decode, what they're thinking or text. And so here, these are just results kind of showing that, these are the baseline results, so the classification error is over 15%. Then training with 10 sessions worth of human data, so just showing multi-session training can be done in this case to really drive down the classification error for these different, the textual decoding. And then here what we're showing is now, what if we take this pre-trained model with 100 sessions worth of monkey data and now fine-tune it and incorporate the 10 sessions of human data with it. And, this is pretty exciting and, perhaps a little bit crazy to think that we could take data from monkeys doing a completely different reaching task, and that can actually benefit our ability to decode from the human brain. So this is really exciting preliminary work. Okay. How long do I have? I forgot when I started. What? 25 minutes. Okay, cool. Because a lot of times I have a lot of questions in between, so then, it's just kind of spreading out. Okay, so I'll talk really slowly. No, I'm just kidding. I have more. Don't worry. Okay, so we have, the results that I've shown so far have been focused on relatively, it's kind of similar brain regions from motor cortex. And so we were excited to think about whether or not we could extend this to, visual cortex, visual tasks, something very different from motor, as well as understand whether or not we can, pre-train on very heterogeneous data sets that might come from different brain regions, as well as different Cree lines or different cell types. And so in joint work with Blake and his student Crystal, we have been working on extending the POIL model to now work across these kind of diverse data sets that are coming from calcium imaging recordings. In this case, a really large data set from the Allen Institute, more Allen Institute love. And so here we're using the kind of same base POIL model that I've described, but now we've extended it to, work with calcium data, so we can also work with regular time series within the same framework. And then importantly, we also have extended this now to have multiple tasks that can be trained multiple tasks that can be decoded during training. So here, I'm just showing a kind of summary of the different decoding results. So in this case, we are training on 10 different tasks. So this is, sorry, I hadn't already said. So in the Allen Institute's visual coding calcium data data set, animals are presented with different types of visual stimuli, things like natural images or natural movies, as well as more synthetic stimuli like drifting gratings of different orientations. And at the same time, they're also recording things like their pupil location as well as their running speed. So you can see how all of these different pieces of information are really kind of diverse in terms of their format. If you want to decode the stimulus information, this is a discrete and sequence level task, versus if you want to decode the pupil or the running speed, these are kind of continuous signals as well. So within this model, we can successfully train on all of these really different types of diverse tasks. It also allows us to now incorporate more data into the model because, if it didn't satisfy the first task you trained on, now you can just kind of augment it and continue to train with further tasks. And so what we find is that, this multitask training on average, does really boost the performance of just the single task model trained on Pollo. And then some really cool things that came out of this were not only just showing that we could unify all these really different data sets that are recorded from different visual areas, as well as different Cree lines, but we also found that there was some interpretable or meaningful information embedded within the latents that can help us to tell the difference between different brain regions that it could have been recorded from, as well as detect or predict the underlying cell type or Cree line that the data was coming from. So these are just some results showing our ability to decode the brain region that the recording was coming from. And this is, baseline model, and then Pollo just trained on a single task versus Pollo trained on all of these tasks. So this is cool. It's like, yeah, we saw that there was some improvement in the task performance, but what we're also seeing is that by incorporating more and more diverse data, we're able to understand things like brain region a lot more effectively. And this is kind of understood, like there are certain brain regions that kind of respond similarly to these simplified stimuli. So once we add in the full naturalistic repertoire, we're seeing this really enhanced performance in terms of decoding from all of these different 6 brain regions. All of them are in visual, but we have primary as well as 5 other kind of higher order visual areas that we're recording from. And we also did a little bit of, kind of digging into whether or not the organization of these latents kind of have some meaning, and found some differences between like lateral and medial regions being organized. And we can do a similar thing now with cell type or Cree line prediction. And again, we found that the ability to train on many tasks is really critical in our ability to see these differences between cell types. And so here's just the confusion matrix. You can see we have 3 different inhibitory lines, as well as, I forget, like on the order of 8 to 10 different excitatory lines. And in previous approaches that have tried to tackle this problem with supervised approaches, like training on the Cree line or the cell type that you want to predict, the performance is relatively capped. And then we do this training on decoding different visual stimuli and see that we can really start to reveal these interesting aspects of the organization of these different cell types. And again, we have done some analysis here to kind of look at the hierarchical organization of all the cell types. We found, broad classes like excitatory and inhibitory being nicely preserved, as well as kind of underlying information about the different projection classes also being kind of captured within this hierarchical structure. Okay, so I hope that I've already convinced you that this multi-session sort of framework can be really powerful and provide us a way to scale and to get information out of data that otherwise wouldn't be possible. But of course, we know that in order to really scale this idea, to go even further, we will also need to be able to do self-supervised training, right? And that's kind of one of the core foundations of a lot of modern models as well. And so, there's a number of projects across the collective groups to try to also apply this with mass models and with other unsupervised training. I just wanted to tell you quickly about one approach that we've also been exploring to be able to use contrastive learning to build representations of neuron level embeddings. So these questions that we have about, cell types or kind of core underlying features that might help us to differentiate different neurons is something that I think is, of broad interest in the field. So this is just one example of how we can use this same like spike tokenization and this same framework to now build these representations of different neural units. We do this through using a contrastive loss. And so the basic idea is just that if we have a large, recording of neurons firing, what we'll do is we'll just use the fact that nearby points in time from the same neuron should be mapped to kind of similar points within our latent space. And so what we'll do is we'll take the embedding for a neuron from two different views, we try to bring those closer, and then we can say that all other neurons could be dissimilar to the representations of a neuron. So this gives us a way of building potentially like a stable view of what a neuron's doing, where it's from, or even its cell type. And in some preliminary work, we've shown that this type of contrastive learning method can be used to also decode cell type. So this is just showing now using this kind of contrastive learning, now we freeze the model and we try to see how well we can read out cell type just from the latents with a linear layer, right? So we're not passing it through a more complex decoder. And what we found is already we're beating, state-of-the-art baselines for cell type prediction considerably through these unsupervised methods. And we can get, a really strong kind of initial performance in terms of these inhibitory types and being able to classify them. So we're excited about, these types of unsupervised methods, as well as being able to move towards like next token prediction and some of these other unsupervised losses. Okay, so in summary, I hope that I've convinced you that scale matters, that we perhaps have been operating in kind of underpowered regimes in neuroscience, and through this kind of multi-session pre-training, we might be able to start to tap into some of the underlying principles or information within these data sets in a completely new way. I also shown some examples of how these pre-trained models can be transferred across different animals, across different tasks, as well as even species. And in the end, we kind of showed a few examples of how these sorts of approaches can also be used to reveal information about brain regions, cell type, and we see already some signs that scaling is also important there, right? Like seeing more animals and seeing more representatives from these different cell types can be super impactful. So this is part of a kind of bigger vision and large collaborative project to build foundation models for neuroscience. We really are excited about the possibility of being able to do these across many different types of tests or brain regions, as well as across different species. We have some preliminary evidence that you can train models both on electrophysiology and calcium jointly. So maybe there are ways to even have multiple types of neural data that could serve as the input, and all this could be fused together. And then in terms of downstream tasks, I've already talked a bit about, the applications to BMI, right? Like being able to build better brain decoders. And some of these ideas of cell type identification, which is a really important problem for the field. But we also think that this could be a really powerful way to almost have like a lookup, right? So by compiling and aggregating more and more brains and more neural data and activity, we'll have a more comparative view, right, of how different animals might differ in the way that they solve underlying tasks. And so having this as a resource for people to understand, like, oh, how does my data set relate to this larger corpus, I think is also a really exciting downstream task and application that we could explore. So this can have, really big impacts on neuroscience. I think I've already kind of said a lot of this, like this unified data analysis, this ability to do comparative inferences across data sets and labs, and then also, with a really rich foundation of what a healthy brain looks like, we might be able to tackle questions about disease in a way that we haven't been able to in the past. So there are, of course, a lot of open questions. In a smaller seminar, like I would hear a lot of these already. So I'll just kind of put them out there and we can think about it. So already we've kind of seen this, we've seen this fact that like some data can be more helpful than others, or some data provides a more powerful source for transfer than others. And so there's a lot of really interesting questions about like what data will be the most critical. Do we already have enough data? How much data will we need? Do we need more? What will that look like, right? So these are questions that we're gonna have to come together as like a community to really face. And, ideally, or hopefully in the end, right, like we would be able to, through this unification, start to see principles of neural computation that are common and shared across all of these recordings and individuals. And so there's a lot of really interesting and exciting work that will go into how we're going to actually find these insights into neural computation within these large-scale models. Right now, they're kind of black boxes. We don't exactly understand what's going on, but already we've seen they're capturing meaningful information that we can pull out of them. So thinking about how do we use foundation models to do science and to understand brain function, like these are really exciting open questions. And then, what is it that the model has learned that allows it to transfer? Like, is it something about the overall statistics of neural firing? Is it something about the correlations or populations of activity that are even across monkey and human, like shared enough to where it can actually effectively transfer? So what are those things that kind of allow this pre-training to really work effectively? And with that, I will just acknowledge all the amazing folks that made this possible. So Mehdi Azabu has really been the driving force of a lot of this in my lab. And now we've been, excited about a collaboration with a lot of folks here at Mila. So Guillaume and Blake, Patrick, Matt, and their students. And really, building these foundations models is going to take a lot of people coming from a lot of perspectives. And so I think this is really the time for us to all, kind of unite. And thank you all for your attention.
Thanks, that was great. We definitely have time for a few questions, so take it away.
Hey, very cool stuff. I was wondering of really the tokenization. So for the spike timing, you of course have the natural just time that you can embed, like with relative embeddings or whatever. Mm-hmm. Like for the units, it's way less straightforward of what you should take, right? Like
of what? Sorry.
Like what the unit embedding should be. Like, do you mostly just want to be able to sort of distinguish between different units that just it's not the same, or do you also really want to incorporate more information? Let's say ideal case, you have a full connectome of the brain that you're also recording from. Like, are there some ways of also including that information it that you actually can generalize better to new subjects and all these things?
No, it's a great question. So in general, you can just have random unit embeddings for a small enough number and it'll still work because it just needs to know that all these neurons are distinct. But then as you start to scale, this alignment kind of forces the model to, or our hope is that it starts to force these things to not be just arbitrary, vectors in the space, but it does start to kind of organize in this way. But then to your other question, we have started to test some of that, like in the case of the calcium imaging, where you can have a lot of other information, like the coordinate, like where in the brain, or the Cree-line information, these other things. And what we found is that for small scale experiments, it did sort of help. But then when you scale up, kind of that extra metadata wasn't It didn't help in terms of performance. Whether or not it could be helpful in reorganizing the space, I think that would be, an interesting thing to test.
Okay, I see. Super interesting. Thanks.
Thank you.
So I didn't keep track of the order, but we're just going to alternate. So, yeah.
Perfect. Hi, thanks so much for a really great talk. I share your enthusiasm. I'm totally bought in on the excitement of this. And so I just wanted to ask, as you mentioned in your open questions, there are a lot of research areas really to focus on, but I wanted to dig into one of them, given that you're so much further along in thinking about these things, which is the kind and amount of data that we really need. So if we're thinking in a fine-tuning context, for example, if I want to take a foundation model like this and go to a very particular data set, I really liked the cross-species example that you gave. Can you give any kind of insights about exactly how much fine-tuning data it seems as though you need at this point? And maybe does that really differ by how far of a transfer you're really attempting to do?
Yeah, I wish I had more concrete answers. It's kind of been weird because, so one result I didn't show here was, but from the Allen Institute, we tried where we would just pre-train on one brain area and then try to transfer to others. And one might think primary visual cortex might be the most helpful at transfer, or maybe if you just have more data, that should be the best. But we actually found some kind of weird things, like certain brain regions were kind of the best at transferring everywhere else. And understanding how and why that was happening is still, very unclear. Is it that there's enough variance in that recording? I guess the one idea would be that you want to have enough variability so that you can kind of,
Project.
you'll see something similar on a testing data set. But there could also be things like in LLMs where there's noisy data, or things where you want to throw away certain stuff that's kind of non-coherent in some way.
Yeah.
So, I think understanding which sources will be most beneficial or what about them is still very unclear. But in terms of the amount of fine-tuning data, I guess we haven't seen a strong effect on how long it takes to train or the amount of shots yet, but it's a good question. I'd love to talk more.
Yeah, no, thank you.
Too. Thanks.
Yep, back to the middle.
Hi, Eva. Thank you for this nice talk. And I have a question about the kind of some monkey to human transfer. So, is this experiment result can be changed into that monkey and human share the same motif? Because their evolution have this kind of very same ancestor. So, is it possible that you can just claim a monkey and a human share the same motif?
I see. Yeah, I guess we have to test transfer to mice to to confirm a very different species.
Yeah, right.
I think there's a number of things that are common between the two. So they're both using the same type of recording device, Utah array, and also in motor cortex. So I guess one potential conclusion would be, yes, there's enough kind of shared structure such that this works, but I don't have a clear answer to that they have similar motifs, right? That would be awesome.
Okay, I see. And I have two more questions. One is that you just had the experiment with Utah arrays. It's very similar to Spike. And are you planning to do the same experiment with something modality like EEG or fMRI, the stuff that can be easily obtained from human?
Yeah, this same approach and ideas can be applied easily to time series data. We have already applied it to calcium, and we also do have some projects looking at intracranial EEG as well. So I think so far we found that the general base architecture is very flexible. So I hope that it can be.
Okay, I see. And final questions. And I saw that in the confidence matrix that you take out the clear lines in the latent embedding of Boyle Plus, the confidence matrix. I see that the task for the inhibitory clear line is much better than excitatory in one. So I'm just curious why it could happen. Do you have some kind of a potential explanation about it?
For
Yeah.
here. So actually, like, there's been a lot of work to look into the differentiability of these cell types using different methods, typically supervised. But we already know these inhibitory lines are much easier to be able to tell the difference between, even with simpler methods. But then the excitatory lines are quite hard. And so the fact that we're getting this amount of differentiation, even of excitatory lines, is kind of, it's kind of cool, or it's at least, I think, a pretty significant result in that sense. But yeah, that the cleanness of inhibitory is kind of already understood that they're quite different.
I see. Thank you so much.
Thank you.
Laura?
So awesome talk. Obviously, it's like such a powerful tool, and I think BCI is like such the perfect framework for using it for like its most effective way of like being able to predict things. But you talked in the end a little bit about how like reverse engineering the networks is like going to be an interesting future direction for science. And I feel like specifically in the context of these behaviors, like the BCI type behaviors, where there actually is a lot known about the dynamics underlying these behaviors, like during preparatory activity, you sort of like set an initial condition, and then you have these rotational dynamics during actual reaching. Like, is that something that you can go in and look in the look in your models and see the extent to which that is the structure that is being utilized across the participants, and like really understand like what is shared? Or is that something that you don't have access to because it's not a dynamical systems model? Or like curious to hear like how you're thinking about uncovering this underlying shared structure across the different data sets.
Yeah, no, it's a great question. So even though we don't have a dynamical systems model, the fact that, so in the latent space, it's still kind of temporally organized. So it could be that, you could still from those latents be able to like plot out certain trajectories over time. But I do think that it would be probably enhanced by, in some ways, us going down to the latent space kind of obscures some of the things that we might be able to see about like the actual timing and certain information of the dynamics if we were to stay with the high dimensional. But yeah, I think in the same way we can do any latent analysis within a deep learning model, we could potentially do those same things here.
I wonder if you were to look at the tokens, if you could like see by the structure of tokens, like if that like, in one token you have like a rotation from some initial condition or something like that. I would be curious to dig into that part
Yeah,
of it.
no, it sounds like a really interesting way to go forward. Yeah, I think we have, been looking at like the unit embeddings or these like learned tokens to kind of see if there's something interesting going on. But I think, to get these temporal dissections, I think it'll, require a bit more thinking. Thank you.
Kareem?
I had a quick question that relates to systems neuroscience, and I was wondering, so in using these models and in moving forward, it seems that there's some consensus that multimodality is going to be important, that we'll be not only having foundation models, let's say, for spiking activity, for LFP type data, ECOG, EEG, MEG, and calcium imaging, and so on and so forth, that we would like to combine them into foundation models that are multimodal, right? Mm-hmm. And so my question is, and this relates to a discussion we had today in the panel discussion about how can these foundation models potentially be useful to neuroscience and bring new knowledge beyond their downstream applications of like better classifiers or better predictors, actually teach us something about the brain. And so there are many open questions in systems neuroscience. How do you relate oscillations to spiking activity? What's the link between gamma activity, high frequency oscillations and things like that, and spiking? So in building these multimodal models, do you see an opportunity there for us to enhance our understanding of the link of multiple observation levels and spatial scales, temporal scales of brain signals? Is that going to be maybe one of the outcomes that, because we need to rethink how we're going to tokenize these things on different types of data? When they work better, because we've combined different modalities, does that tell us about the complementarity between these signals? If they don't work better, maybe it's just because they include the same information.
Yeah, it's a great Great questions, and like for sure, multimodal can, give you kind of complementary insights. So I've been talking mainly about like, oh, I'm gonna stitch together all of these recordings because, electrophysiology is very limited and it's only gonna give me this much. And so I would have to combine them all together in this way. But then the reality is like, you'll never see all of the brain, right? And you'll have to go to some other modality to see things at a different scale or to see those oscillations. So yeah, it's probably not enough to even stitch together all of the electrophysiology. Like you have to see the brain at many different resolutions probably in order to put it all together. And I kind of also think that some of these intermediate modalities can kind of serve as stepping stones, right? So it's like, LFP to EEG or LFP back down to raw spikes. So yeah, I think probably we will have to be doing things across many different spatiotemporal scales in a multimodal way. I think the challenge often with doing like paired multimodal training is often you don't have both of the modalities simultaneously recorded. So I think we'll have to have ways to leverage that when we have it, but then also train on things kind of individually and just. Because I think the other part of it is like to get to scale, you want to make the fewest assumptions about what you expect to get, right?
Thank you so much.
Thanks.
So I think we've been at time for one more. What's the schedule finally?
Okay.
Yeah, sorry, maybe like I just missed it. I'm particularly interested in how you deal with individual spikes. Do you encode the timings and treat them as point processes and encode relationship, or you encode firing rates?
I'm trying to remember.
And just try to get to input-output description of the whole system. I'm just curious.
Yeah, I wish I had my slide here with the equation, but basically this is, so within the transformer you have this notion of the token, but you also have its position. So position embeddings kind of provide an important role. And so often when you have a sequence, they'll use absolute position embeddings like token 1, token 2, right? Like, and so on and so forth. In this case, we're using something called relative position embedding, which basically will, for each pair of spikes, or each 2 tokens, you compute the distance between their spike times. So it's like Ti minus Tj. And then that absolute difference is then fed into the attention calculation. So instead of just normally having your dot product between your keys and your queries, you're also kind of multiplying that term or the attention score by this output of this relative position encoding. So I guess the best way to say, or at a high level, we are able to take the relative difference between spike times and incorporate that into the sort of training procedure.
Okay, so this level of individual spikes, so why not using firing rates?
So there's a few reasons. One is we think that, perhaps, in some cases, when you bin, you can lose temporal resolution, right? Like you don't know exactly when that spike occurred. That could, in many cases, smear information. So maybe when you're trying to form associations, you think that all these things happen at the same time, but one actually happened a little bit after the next, and that causal interaction is kind of important. So one is like, maybe in many cases, spike times might truly matter, so let's keep them rather than throwing them away. And then the spike times matter, and then the other thing is like when you're dealing with data from different brain regions and from different stimuli, the bin size can be quite different. So now if you're dealing with aggregation of all this data, you have to start making decisions about how you're going to bin. Maybe I chose too big of a bin here, too small of one there. So it also gives us this kind of flexibility that I think is important. And we don't have to store tons of zeros, right? Like computational efficiency is probably also part of it too for scaling.
Thank you, like amazing model. Thank you very much.
Thank you.
All right, one quick question, and then we'll.
I just have one quick question about the model. So I believe you use the perceiver I/O model to kind of I believe you use the perceiver I/O model to map the different inputs so that the model can be both multitask and multimodal. So I was just wondering because, like, for example, like in language modeling, they use different types of multimodal methods to kind of group together different modalities and tasks. And was there a reason why you chose perceiver I/O specifically, or have you tried different integration methods?
Okay, sorry. So so have we tried? So before this, like we had been doing a lot of work in using self-attention models or kind of vanilla transformers on small data sets. One of the things that came out of that was the significance of actually tokenizing each channel independently. So previous transformer models for neural data would actually tokenize all neurons jointly. So you basically take all of your neurons, you slice through for a given chunk of time, that becomes your token. Now the problem with that is that depends on now the ordering of those neurons. And so this doesn't generalize to a new data set. Like if I even just reorder them, I'd have a completely different representation. So the first thing we realized, you need to tokenize at the channel level. But then you have so many tokens, it's actually quite hard to do things effectively. So the perceiver is also an important compression mechanism. And there have been some other attempts of some state space models amongst the group too. So I think that there certainly can be benefits from other types of architectures, but this is one that kind of fits the bill and has a number of important aspects that seem to really make it quite performant and flexible. But understanding where all of that benefit comes from, I think, will be important in the future.
Great, thanks. Let's thank Eva again.
Thank you.